Welcome dear students. This is my cellular energetics lecture series. In this video we are going to learn about TCA cycle. This TCA cycle is very important in our metabolism. The TCA cycle is also known as Kreb cycle and citric acid cycle. So today's topic is TCA cycle. Or you can remember Kreb cycle or citric acid cycle. The naming of TCA cycle, the full form of TCA is tricarboxylic acid cycle, and the name Kreb from scientist Hans Adolf Kreb, who had discovered this pathway, actually worked on this pathway and was rewarded by Nobel Prize in 1953. So this is also known as Kreb cycle. And the citric acid cycle, because the first product of this cycle is the citric acid. And in the citric acid there are 3 carboxyl group are present, 3 COOH group are present, so it is called tricarboxylic acid group. Now let me quickly summarize what is happening here. From the glycolysis, one glucose molecule is actually converted into pyruvate, we know, and this thing actually occurs in the cytoplasm. Now we all know that glucose is actually a 6 carbon molecule and the pyruvate is 3 carbon molecule, so there should be 2 pyruvate. Then the pyruvate enter into the mitochondrial matrix and is converted into acetyl CoA. Now there should be 2 acetyl CoA, and the acetyl CoA is actually 2 carbon molecule, so 2 carbon dioxide actually is released in this process. I have already discussed in my previous lectures. Now the acetyl CoA present in the matrix, another molecule actually present in the matrix called oxaloacetate, or you can remember it's oxaloacetic acid. This molecule actually attached with acetyl CoA. I'm not writing here the 2 because the oxaloacetic acid or oxaloacetate actually associate with one acetyl CoA. So the cycle actually runs 2 times to actually convert the 2 acetyl CoA. Okay. The first step in this reaction is called aldol condensation. Remember each and every step very carefully. It is very easy to remember. There are 9 steps, and in these 9 steps there are 3 steps that are irreversible and the 6 steps are reversible. So carefully watch this video, each and every step, that is very important. So in the first step, oxaloacetic acid already present in the matrix associate with acetyl CoA, this is called aldol condensations, and make the first product of this cycle called citric acid or citrate. So that is why this cycle is called citric acid cycle. Now the enzyme that actually convert these 2 molecules into citrate is called citrate synthase. Very simple, you can remember. Okay, the name citric acid because this is the first product, is the first reactions, so it makes citrate. So which enzyme will make the citrate from this product? Citrate synthase, that can synthesize citrate. Now I have shown here this process is actually irreversible process, so citrate cannot be converted into these 2. So this enzyme should be allosterically regulated. Now ADP can activate this molecule and ATP can inhibit this molecule. Also the substrate citrate or citrate can inhibit this molecule. Okay. Now in the second step, you remember this thing that isomerization occur. Isomerization. So the atoms arrangement should be changed in this way. What happens here? At first the citrate is converted into cis aconitate. This is the intermediate product of these isomerizations, and then actually this step is actually reversible step. So let me in a reversible manner, and the cis aconitate is actually ultimately converted into the isomer of this citrate, and the isomer is called isocitrate. Very easy, isocitrate. So this step is actually occurring in 2 and 3 step. So this is step 2, this is step 3, and the same enzyme actually convert citrate to cis-aconitate and cis-aconitate to isocitrate is called aconitage. Okay, aconitage. The second and third step actually performed by an enzyme called aconitage. Okay, so this step is reversible. Okay, now what happens here? One water molecules actually come out in this step, and water molecules actually enter in this step and actually rearrange the citrate molecule to isocitrate, so it is called isomerization. So remember, first step, these two molecules associate with each other, make citrate, it is called aldehyde condensation. The enzyme is citrate synthase. This is the first step. In the second and third step, isomerization occurs. The citrate ultimately becomes isocitrate, it is called isomer of the citrate. Just the atoms rearrangement occur, and in this step there is the intermediate product called cis-aconitate. Both the step 2 and step 3 actually performed by aconitage. This process is reversible. Now another thing occurs in the fourth step. This isocitrate is actually go through a irreversible process and make another product called alpha ketoglutarate. You can remember this step easily, alpha ketoglutarate, and this step is actually irreversible step. So this is the second irreversible step. So I can write here this is the fourth step, and in the fourth step, so this is the second and third step. In the fourth step, the reaction is called oxidative decarboxylation. Okay, oxidative decarboxylation. What does it mean, oxidative? Oxidative means in these reactions there should be an oxidation and reduction reaction, and decarboxylation means one carbon dioxide will be released, because we know in my previous lecture I have mentioned that from the pyruvic acid to acetyl CoA carbon dioxide release, so it is also known as decarboxylations. So decarboxylation means one carbon dioxide will be released. Now let me write that this citrate is actually 6 carbon molecule. This cis-aconitate is also 6 carbon molecule. Isocitrate 6 carbon molecule. Now in this step 1 carbon dioxide is released, so the alpha ketoglutarate is actually 5 carbon molecule. Okay, this is a 5 carbon molecule. The name of the enzyme you can remember very easily. This name is, the enzyme name is isocitrate dehydrogenase. You have to remember this thing. Let's look at this. Isocitrate is converted into alpha ketoglutarate, and the enzyme name is isocitrate dehydrogenase. I have mentioned this is an oxidative decarboxylation. Decarboxylation means carbon dioxide will be released, and this dehydrogenase, this enzyme dehydrogenase means it will exchange hydrogens. To which it will exchange the hydrogens? NAD plus will exchange hydrogen with this and make NADH plus H plus, and this NAD plus actually get reduced, and isocitrate is actually oxidized to alpha ketoglutarate, so it is called oxidative decarboxylations. And the enzyme you can remember very easily, the isocitrate dehydrogenase. This is the irreversible step, so this enzyme should also be regulated. Yeah, this enzyme can be regulated by or activated by calcium and ADP. Okay, both these molecule can activate, and if there is high level of ATP, it will inhibit this product. Also high level of NADH inhibit this enzyme. Okay, now the fifth stage is also a irreversible step. This is the fifth step, and the fifth state is also a oxidative decarboxylation step. What does it mean? Decarboxylation means one carbon should be released, carbon dioxide, in the manner of carbon dioxide. So the product that is produced here is called succinyl CoA. So you can see here that the alpha ketoglutarate was 5 carbon. Now the product release 1 carbon, so it should be 4 carbon molecules. Okay. Now here also oxidation occur, decarboxylation occur, and the oxidation actually, that means, yeah, the name of the enzyme you must remember, that is alpha ketoglutarate dehydrogenase. Very simple. You see here that when any product is produced, the name of the enzyme is actually go through the product, like oxaloacetate plus acetyl CoA producing the citric acid, and this is called citrate or citrate synthase. Now here aconites don't actually match with this name, but you can see here isocitrate to alpha ketoglutarate we call it isocitrate dehydrogenase. From the alpha ketoglutarate to succinyl CoA we call it alpha ketoglutarate dehydrogenase. Okay. Now the dehydrogenase actually exchange the hydrogens with NAD plus and release NADH plus. Here you can see succinyl CoA produced, so that means CoA actually added here, coenzyme A actually added here. Now this is also, this step is also irreversible step, so this alpha ketoglutarate dehydrogenase should be actually controlled by allosterically. So same thing, the calcium can activate, ADP can activate, but ATP and NADH will inhibit this enzyme. Okay. So up to that, the succinyl CoA is a 4 carbon molecules. Now this succinyl CoA actually go through a reversible reactions and produce succinate. Very easy, succinate or succinic acid. So this succinate should be 4 carbon molecules, and here one thing, very important thing occurs, coenzyme A actually released, and by that time GDP plus inorganic phosphate actually associate to make GTP. So here one important, because we know that this NADH is very important for electron transport chain, I have already written here, so we will count this NADH. And GTP is also important because the GTP is ultimately converted into ATP. How? Because GTP and ADP exchange the phosphate each other and make, this is also a reversible mechanism, make GDP and ATP. This enzyme is called nucleoside diphosphate kinase. This enzyme can perform these actions and make ATP. So we will count as a ATP, not GTP. Okay, now succinate. Okay, now I have not written the name of this enzyme. So this is the 6th step. The 6th step is actually a cleavage step in which the CoA actually cleaved out, and the enzyme name of this reaction is called succinyl synthetase. You can see here a difference. I have mentioned that this is a citrate synthase and here is a synthetase. What is the difference between synthase and synthetase? The difference is here, in synthetase always there should be some ATP or GTP destructions. But you can ask me why there is, because there is from the succinyl CoA to succinate, GTP is actually producing. One GTP is actually producing. So why it is called synthetase? Because you have to understand this, because this reaction is actually reversible. So succinate can be converted into succinyl CoA. So that's why it's called synthetase, because when succinate is converted into succinyl CoA, it produces GTP. The reverse, just think about the reverse, because succinyl CoA to succinate is actually destroy the convert it into GTP. But succinate to succinyl CoA, it will degrade the GTP. The reverse will occur. Okay, so this is called synthetase, because in synthetase enzymes, one ATP or GTP will be cleaved to do the reactions. So this is the 6th step. Now from the succinate to it produce a product that is actually reversible. This step is reversible, okay, and produce fumarate. And in this reaction, this is actually called the seventh reaction is called oxidation. Here oxidation occur, and what happens? A FAD is actually converted into FADH2. You have to remember this. FAD is converted into FADH2. This is the oxidation step, and the name of the enzyme is succinate dehydrogenase. Succinate dehydrogenase. The specialty of this enzyme is that this succinate dehydrogenase is a subunit of succinate CoA reductase. This actually enzyme is the subunit of a enzyme complex called succinate CoQ reductase. This is actually complex 2 of electron transport chain. Okay, now this enzyme converts fumarate. This fumarate is also 4 carbon molecules. Now another time reversible reactions occur. And the fumarate make malate. Okay, malate. So this step is 8 number step. The enzyme name is called fumarate. Okay, this step is known as hydration step because here one water molecule is actually added to make malate. This malate is also 4 carbon molecules. Now the malate is actually converted into oxaloacetate. This is also a reversible step, and the name of the enzyme, this is the last step, this is the 9th step, and it produce actually the enzyme name is malate dehydrogenase. Now I think that you can understand where there is a dehydrogenase, that means it rearrange or actually exchange hydrogen. So this step also produce NAD plus to make NADH. You have to remember this. So you can see here this cycle is very easy to remember, but you just need a focus. So let me repeat this again. Oxaloacetic acid or oxaloacetate, OA, always there in this mitochondrial matrix. Acetyl CoA is produced, it gets aldol condensations and makes citrate. The first product so is called citric acid cycle, and it contains 3 carboxyl groups, it's called tricarboxylic acid. Citrate, this step is irreversible, so I'm marking star. This step is irreversible, so this enzyme should control by allosterically. Now in the second and third step, isomerization occurs. Simply the citrate becomes isocitrate, isomerization occur. Okay, and then enzyme name is aconitage. Now in this step it is actually isomerization. In the fourth step, first oxidative decarboxylation occur. Decarboxylation means carbon dioxide will be released. Oxidation means here oxidation and reduction occur. NAD plus is actually get reduced, and the isocitrate gets oxidized to alpha ketoglutarate, and it becomes 5 carboxyl. This step is also irreversible step, so I will make a star mark. So this enzyme can be regulated. In the fifth step, the name of the enzyme is alpha ketoglutarate dehydrogenase. You remember this name very easily because the name of the substrate, previous substrate actually gets the name of the enzyme, alpha ketoglutarate dehydrogenase. Make succinyl CoA. Now the succinyl CoA makes succinate. This is actually a cliff product because coenzyme A here a coenzyme may actually get attached with the alpha ketoglutarate to make this succinyl CoA, and then it actually release the CoA to make succinate, and the enzyme is succinate synthetase because they use ATP or GTP. You can see that here a GTP is actually producing, but the reverse can be occur, and in that case the GTP actually get destroyed into GTP, so it's called synthetase. Now the succinate actually make FADH2. Actually I am telling you that this enzyme is very important because it is actually part of the complex 2 of this ETC, electron transport chain. It produces FADH2. We also count this product. Now fumarate, it produces fumarate. Fumarate to malate is called fumarate. From the malate to oxaloacetate, malate dehydrogenase, because it actually exchange hydrogen. So in this cycle we have got very important some products that we will count here. Like we now write here that from the 2-acetyl CoA 4 carbon dioxide will be released in this TCA cycle. This is the TCA cycle. 4 carbon dioxide will be released, and it will make how many this product that will actually help in the electron transport chain? We count it. The first NADH, here is the first, here is the second, here is the third. So there are 3 NADH is producing from 1 acetyl CoA. So from 2 acetyl CoA we will get 6 NADH. This is a electron carrier, high energy electron carrier. Now we also get FADH2, so we will count it 2 FADH2, because from 1 acetyl CoA we get 1, so from 2 we will get 2 FADH2. We will also get 2 GTP, that is converted into ATP, so we will count it as a 2 ATP. Yeah, so this is the net gain of this reactions.